All right, ladies and gentlemen, welcome back. We are going to continue talking about your adaptive immune system today. And this response is really characterized by the specificity, right? It's specific to the pathogen that it's encountering. It's capable of maintaining memory, but that means that there's going to be a lag time in the amplification of this defense in ramping it up. This system also needs to be able to recognize self versus non-self, being able to recognize and have what we talk about as self-tolerance. What are the cells that belong to you? What sort of condition are they in? Do I need to get rid of these cells because they're worn out, they're abnormal, they're mutated, they're cancer cells? Is this an okay cell because it belongs to me and it's healthy, right? All of those sorts of things. It is also a systemic response. So we're going to see these cells working throughout your body as opposed to just being potentially localized, like we might see in the innate immune response, where they are going to that initial site of infection and just kind of hanging out there. And it's also very much genetically controlled. So there are genes that determine what belongs to you, what is foreign, how do I recognize something foreign and what it is. And so this antigen determination is all genetically controlled within the cells that are part of this immune system, or this section of your immune system. So let's talk through some basics here. So the cells of your immune system, both innate and adaptive, are circulating throughout your body, mainly in your lymphatic system, so your lymph ducts, your lymph nodes, as well as your blood. And when pathogens get into your body, your innate immune system initially goes to that site of infection, wherever that happens to be. So if it's a puncture wound, they go there. If it's an inhaled pathogen and it's sitting in your lung tissues, they're going to go there. And those innate cells, those non-specific cells, are going to take care, or at least attempt to take care of those pathogens initially. But in the process of doing that, a lot of things like your phagocytotic cells, when they take in those pathogens and attempt to rid your body of them, they're also going to recognize the antigens that are present on that pathogen, in the sense that they're going to just take the random antigens and present them, put them on their cell surface, and take them to lymph nodes where B and T cells that are part of this third line of defense tend to hang out. And so we're going to get to these lymph nodes, and that's where a lot of this initial detection from your adaptive immunity takes place. So let's talk about those cells that are part of your targeted defenses here, the ones that are going to specifically recognize invaders and be able to distinguish which invaders they're dealing with. So these are your lymphocytes, your B and your T cells. And both types of lymphocytes originally developed from stem cells in your red bone marrow. Once they get past that initial immature stage, then it's a question of where they end up. Do they stay in your bone marrow, or do they get transferred to your thymus? And that's where these distinguishing pieces of your B and your T cells will sort of diverge from each other. So those immature lymphocytes, if they stay in your B cell, if they become your B cells, they will continue their maturation stage in your bone marrow. If they go to your thymus, then they will become your mature T cells. Both of those will then go to your lymph nodes. Once they get to those lymph nodes, they're still what we're talking about as immunocompetent, but naive. So they have matured to the level necessary to be capable of recognizing different antigens, but they haven't fully completed their task yet, if that makes any sense. So sort of think of those immature lymphocytes in the bone marrow as baby cells, they get to your bone marrow to finish that recognition, or they get to that thymus to finish their recognition to kind of get through high school. Once they get to those lymph nodes, that's where they're sort of in college and waiting to finish their training to start a career. And that training, if you will, to get to their official job, their college education, if you will, is going to only occur once they encounter antigens from a foreign material. So they're just going to kind of hang out in those lymph nodes. You may also see some cycling through blood and other tissue fluid, just waiting for an antigen to come activate them. So bone marrow is where they initially develop. They continue development in your thymus or your bone marrow, depending on whether they will be in T cells, and then they start circulating, waiting to connect to an antigen. So let's dive a little deeper into each type of cell. Your B cells, again, these will develop and mature in your bone marrow, and these guys will primarily defend against pathogens found in your body fluids. So a lot of parasites, bacteria, that sort of thing. Even materials that those pathogens might create, so toxins from bacteria, for example. And these guys are capable of differentiating further, right? Sort of getting through that college education, if you will, into plasma cells. And those plasma cells are a specific type of B cell that secretes antibodies. The only way we develop into those plasma cells, though, is to be activated by a specific antigen. So we need to make that antigen-antibody connection, but that's only going to happen once those antigens have been presented to those naive B cells, say in your lymph nodes or circulating through your tissue fluids. Once they have been activated, they will differentiate into these plasma cells and make antibodies that are specific against the antigen that was presented to them. Once those antibodies get produced by these B cells, those antibodies then themselves can go circulating through your body fluids and start finding those antigens anywhere else in your body. So these guys are involved in that humoral immunity, right? This is the specific immunity produced by those B cells, really by the creation of those antibodies. And those antibodies aren't going to be what destroys the pathogen. Instead, those antibodies are going to attach to those antigens and simply tag them as things that shouldn't be there. They're going to help to neutralize things, but it's going to ultimately take your T cells to destroy the pathogen itself. So if we look, right, here are all of those different naive B cells that are floating around in your lymph nodes, floating around in your blood. And if you take a look, remember I said that there's a genetic component to this. So your genes get expressed a little bit differently in one B cell versus another, which is how we get these different antibody receptors that are sitting on the B cells. Okay, and they're just waiting to find antigens that connect to them. So this antigen from a pathogen, let's just say it's a strain of the flu, when that gets presented to those B cells, it's only going to connect to one particular set of B cells that happen to have the receptor that is correspondingly connected. They fit together like puzzle pieces, or like you would typically see a substrate to an enzyme, right? There's a lock and a key to this. So once that particular B cell that has the corresponding shape to the antigen gets activated, the rest of these just kind of hang out, but this guy then gets activated and starts going through cell division to make more of itself. We'll then work on some additional genetic control mechanisms to go through a set of differentiation, meaning that we'll turn on certain genes, turn off some others, to turn those activated B cells into plasma cells. And then those plasma cells will use their set of genes that are now currently turned on, that were not on back here, to now mass produce those antibodies that are specific to the antigen that activated this set of B cells in the first place. Those antibodies then get secreted out into your blood, into your lymph tissue, into your tissue fluids, and they are just searching to find the antigens that they match. Once they make that connection to that antigen, they'll stick to it, and now they have tagged that bacterial cell or that infected body cell that has a virus growing inside of it, or even potentially a cancer cell. These will tag these things that aren't supposed to be there to now allow your T cells to come in and take care of the problem. So your T cells develop in your bone marrow, but they will go to your thymus because they need to do an extra level of training, if you will, that occurs in your thymus. And that extra training is going to be how it distinguishes your cells from other cells. T cells are going to work directly in attacking host cells that contain multiplying pathogens or that are the pathogen itself, if it's bacterial or some sort of other parasite, or they will go attack cells that are currently growing a bunch of viruses. These are involved with the cell-mediated immunity. Okay. So again, just like those B cells, each T cell has its own receptor for a specific antigen. And they're floating around your body, in your lymph nodes, in your blood, in your tissue fluid, waiting for the antigen that is specific to its receptor to latch on and connect. The other difference here, though, is that your thymus allows these T cells to acquire what we talk about as major histocompatibility complexes. They're your self-antigens. We're going to teach your T cells which cells belong to you, as well as allowing them to have the ability to recognize cells that don't belong there, pathogens that don't belong there. So in order to activate your T cells, not only do they need to come in contact with a specific antigen that connects to them, but they also get a secondary confirmation in terms, or a secondary activation in terms of being able to recognize your cells from something else. So these guys produce these cytokines that are specifically called lymphokines. And these, again, are signaling molecules that your cell creates as a way to coordinate function, because there is no communication in the sense of speech or written language or anything like that. It's all chemical. Those lymphokines are proteins that are used to direct that immune response between signaling cells. So these guys produce these lymphokines as a way to help coordinate all of this targeted defense between the different B cells, the T cells, the innate immune response cells, macrophages, that kind of thing. So they're going to really kind of be the players in coordinating all of this once they get activated. And again, T cells are going to require two signals. They're going to need that initial antigen recognition. All right, so we're going to activate a certain subset of T cells, just like we did B cells, by having their receptors connect to the antigen that is specifically shaped for them. And then they will also produce substances during your innate immune response to microbes as a way to co-stimulate them, to ensure that this really is something that is foreign, that doesn't belong to you, and really does require an immune response because it's potentially dangerous. So this is kind of what we're talking about here. So they'll get that initial antigen connection, but then they'll also get a additional signal from these MHCs, or that are part of things like your macrophages from your innate immune response. So they're going to not only just recognize that that antigen is in existence, but they're also going to need that MHC molecule presented from a macrophage, for example, as a way to say, yes, this is definitely something that we need to take care of. And so here's sort of where this proliferates out. Don't get lost in all of these codes, but you've got those lymphocytes, right? They're going to go from your red bone marrow into your thymus. This is where they're going to get that learning of what those MHC proteins should be. And then when they recognize them on a particular innate immune response, they'll connect not only the antigen, but also the MHC protein. And then they'll start differentiating out from there into some regulatory T cells, helper T cells, cytotoxic T cells. And then these guys will also be able to create some memory cells, just like our B cells will, that we'll see as we move forward. So when we look at your T cells and what they do, you just saw that they break out into different types. Your cytotoxic T cells, these are the ones that are going to go to your body cells that are infected and directly attack them. So these guys are going to be similar to those natural killer cells, except that these are now targeted defense, where those natural killer cells were much more non-specific. So these guys will also produce those perforin proteins, which will go into the cell membrane of an infected cell, punch a bunch of pores or a bunch of holes into them, and cause all of that cellular fluid to leak out, break open, die off. As a result, those cells can no longer continue to be virus factories as they currently are. Your helper T cells are really important because these guys are going to activate not only your cytotoxic T cells, which are going to go attack infected cells, but they will also further activate your B cells to help increase that humoral immunity and the secretion of those antibodies. So those helper T cells are the ones that are directly being connected to macrophages from your innate immune response. So they're going to be the ones that get that initial connection to the antigen, recognize that MHC, and then go out and say, yep, cytotoxic T cells, there's definitely a problem. Here's the antigen you're looking for. Go take care of those infected cells. They'll also then stimulate additionally those B cells to create those antibodies. So these helper T cells are really important because they are the major connection between your innate immune response and your adaptive. So when we get to talking about HIV and AIDS, that particular infection goes after these helper T cells, which means it shuts down everything beyond this. This never happens in those HIV infection patients because those T cells are just gone. They don't exist. And so that communication piece between your innate immune response and your adaptive one is obliterated, which is a really big problem.